Hello and welcome. Let's start talking today about the last major component, basically, of the cardiovascular system before we jump on to lymphatic and immune systems. So, here today, what we'll do is we'll talk about then blood. Blood. Now, when we go through and we talk about blood and basically this whole circulatory system, this cardiovascular system, uh, you can see here now main functions include transport, protection, and regulation. But we'll go through and we'll talk about all these different functions as well. Let's first go through and let's talk about blood. Now, when we go through and we talk blood, blood is going to get divided basically into three major components. So here we could see one image kind of depicting that. Here we could see another image. Here, basically, what we're saying is, if I was to take some blood, you will actually do this in physio. Uh, you'll take blood, and when you remove blood from yourself, you'll fill it into a capillary tube. You won't need so much as they're depicting here. And what we'll do then is you will, like we're seeing here, you're going to get that blood, and you're going to put it into this little tubes, the capillary tubes, and then you're going to spin it in a centrifuge. Then you will spin it in the centrifuge. Now, when you spin in the centrifuge, here what it's going to do is it's going to spin at such a high rate, and what that does, it separates all the components of the blood, basically. Now, when we separate all the components of blood, you can see here, according to weight, basically, is how kind of they're found. So the heavier components are going to be, we can see the erythrocytes, the red blood cells. Now, when we talk about erythrocytes or red blood cells, you can see these erythrocytes or red blood cells are going to make up a certain percent. Same thing with the buffy coat, and same thing then with the plasma as well. So here when we go through and we talk about blood, the composition of blood you can see now includes plasma, buffy coat, and erythrocytes. Now the function of this life-sustaining fluid is that it serves as a transport vehicle. It's going to serve as a transport vehicle. This is the only fluid tissue that's going to be found in the body. So when we talk components, the components of blood are going to be liquid, and they will be also cellular. So liquid and cellular components. Now blood is a specialized connective tissue, if you recall, in which you can see formed elements are suspended in plasma. So when we talk formed elements, what are the formed elements? They are the living blood cells. They are the living blood cells. When we talk living blood cells then, let's go through the living blood cells. Here we could see first we have erythrocytes. Erythrocytes are your red blood cells. And their job, their overall function is going to be to transport oxygen. I mean, we're going to see CO2 as well, and how it all happens, but just speaking overall function, transport oxygen. Next, then you have your leukocytes. Your leukocytes are basically your white blood cells, and they're found in the buffy coat. They're going to be found in the buffy coat. The buffy coat is going to be the thin, whitish layer in the middle. The thin, whitish layer in the middle of the erythrocytes and the plasma. So here we have the buffy coat. Next, then we have platelets. Now we talk platelets. Platelets are going to be cell fragments. They are cell fragments, so pieces of cells that are going to help stop bleeding. That will help stop bleeding. And they're also going to be found in the buffy coat as well. So leukocytes and platelets, and you can see they make up less than 1% of whole blood. Unless, obviously, active infection, so then more white blood cells will be in the picture and that population will increase. Next then is the plasma. Plasma is going to be the non-living fluid matrix. It's the non-living fluid matrix. Remember we talked about different characteristics in relation to connective tissue, and matrix we saw was one of those components that we had here. And the plasma is going to suspend the formed elements. It suspends the formed elements. And it's kind of straw colored, as we can see here. It is straw colored. And we'll talk more about that. Now, when we go through and we talk physical characteristics of blood, okay, so here you can see all the various formed elements we've just gone through and we've mentioned, and we'll look at each of the white blood cells then in greater detail as we progress. And then the backdrop would be plasma. Right here they don't have it. That's why they're not showing it. Plasma is not white. Like I described it, it's this color here you see. So when we go through and we talk about blood, you'll see some physical characteristics I want to talk about. First is the color. When we talk about blood, the color varies, right? It goes from red to being blue depending on our mood, right? If we're not feeling happy that day, blood is blue, right? I mean, I'm in an overall generally good mood, but look, mine is blue today, so maybe I'm not. I don't know, right? Is that the case? No. This is a collagen UV interaction creating that blue color, okay, in relation to the veins. Our blood is going to be red. When we talk about color, the color of our blood is going to vary dependent on the amount of oxygen that it's carrying. Varying on the amount of oxygen it's carrying. So blood can be a scarlet to a dark red, a bright red to a dark red. Bright red, scarlet, if it's O2 rich, if it's oxygen rich. Dark red, if it's oxygen poor. Now, when we talk density and viscosity, we'll compare it to water. Blood is going to be more dense than compared to water. And it's also 5 times more viscous. When we're talking viscosity, we're talking the thickness. It's 5 times more viscous. Now, when we go through and we talk about pH, pH of blood is slightly alkaline. pH of blood varies from about 7.35 to about 7.45. Okay, 7.35 to 7.45. Difference of 0.1. Even that 0.1 is a drastic change. It's a drastic change. The body will know about that change. Now, when we talk temperature, temperature of blood's temperature is slightly 100.4 degrees Fahrenheit. It's slightly higher than body temperature, about 38 degrees Celsius. Now, when we go through and we talk about the functions of blood, the function of blood is going to be distribution. We can see number 1, distribution. Excuse me. Now, distribution, it's going to be responsible for delivering oxygen. Blood is responsible for delivering nutrients. Blood is responsible for transporting metabolic waste, hormones, vitamins, right? We can go on and on and on and on with the various components that are going to be found. So distribution, you can see, is one overall function. Regulation, you'll see blood is going to be responsible, or it'll be helping regulate body temperature, regulates pH, also regulates fluid volume, and it regulates fluid volume. Fluid maintenance you can think of. Protection is the third function. When we talk protection, you'll see blood is going to prevent blood loss, so one form of protection, and blood is going to also protect us from infection. Also, protection from infection, right? Our warriors, the white blood cells we could think of there. So let's talk plasma then. When we talk plasma, plasma is going to be straw-colored, as we can see here. Plasma is straw-colored. It's a sticky fluid, mostly water. It's mostly made up of water. And you'll see it's also going to contain over 100 solutes. We won't go through every single one of them. We'll talk about a few of them. Now, the main component making up plasma is going to be water. Water makes up about 90% of plasma. So 90% of plasma is water. Now, when we talk solutes, first solute we want to talk about here are going to be proteins. Now, when we talk proteins, let's move slides. So here we can see blood plasma, the components of blood plasma. We're going to go through those here now. So we can use this image here. Now when we go through and we talk about proteins, proteins you can see are going to make up, they're going to be, first of all, the most abundant solute. After, we saw the most abundant solute, but the second most abundant component, you can say, right? Because water is the main component. So they account for about 8% of plasma proteins. Oh, I'm sorry, of your plasma. So when we talk proteins, they're going to account for about 8% of your plasma. They account for about 8% of your plasma. So you can see about 98%, bam, taken care of right there. And most of the proteins are going to be produced in the liver. They're produced in the liver. Now, I'm talking proteins produce. Now, this is all biology. You should recall where, more specifically speaking, if I was to ask you inside the cell is protein synthesis going to take place, then is where you should be able to talk about that, and you should be able to talk about the processes, right, that are involved in all of this there as well. They have cute names to them, right? You should be able to review all that and have that down there as well. So here when we go through and we talk about then albumin. Albumin is going to account for about 60%. It will account for about 60% of our plasma proteins. So you can see it's one of the most abundant proteins there. 60% of plasma proteins. And albumin, and what it does, it acts as a carrier to shuttle molecules. It acts as a carrier to shuttle molecules. It's an important blood buffer. It helps buffer our pH. It's a major protein in plasma osmotic pressure as well. Amongst other functions you could see are listed basically there, so make sure you got those down as well. Next then we have globulin. Globulin, we have three types. Now globulin proteins make up, you can see, they account for about 36% of plasma proteins. That's about 96% proteins you can see right there. So globulin, three types, they account for about 36% of your plasma proteins. We have alpha, we have beta, and we have gamma. Alpha, beta, and gamma globulin proteins. Fibrinogen, another protein, fibrinogen. Fibrinogen makes up about 4% of our plasma proteins. About 4% of our plasma proteins. And fibrinogen, you'll see fibrinogen is going to form fibrin threads. It will form basically fibrin threads that are going to help, or that will be involved with blood clotting, that are going to help clot blood. Next then we have nitrogenous waste. Nitrogenous waste. Now when we talk nitrogenous waste, we'll see here nitrogenous waste now will have basically there as well. And these are going to be byproducts of cellular metabolism when we talk nitrogenous waste. Byproducts of cellular metabolism. And nitrogenous waste will include substances such as urea, uric acid, and creatinine, and even creatinine. We have also organic substances, organic nutrients here as well. We talk organic nutrients that are going to be found in the plasma. Organic nutrients will include materials absorbed from the GI tract. It'll include materials absorbed from the GI tract and then transported for use throughout the body. And this will include substances, again, organic, so you should recall from bio as well, carbon is a backbone, right? This includes glucose and other simple carbs, amino acids, fatty acids, cholesterols, right? Cholesterol and vitamins. We also have inorganic ions. You can think of electrolytes, and these include cations like sodium we can see here, potassium we have listed, magnesium, calcium, right, chloride, and various others. We're also going to find respiratory gases, such as oxygen and carbon dioxide, to name the major couple ones. Nitrogen as well. Now, if you really want to see what's in the air and what you're breathing in, I mean, you can go to the EPA reports and see, what's basically going on in your. I think they might have those available for us. And then hormones as well. Hormones. Now, when we talk hormones, steroid and thyroid hormones that are going to be carried by plasma proteins. And then regular hormones are also going to be traveling through here as well, not just the steroid and thyroid hormones. Next, then, let's talk about our formed elements. Let's look at our formed elements. Now, when we go through and we talk about our formed elements, here first, again, we're looking at our living blood cells. So here first, I'd like you to understand, when we talk about our living blood cells, let's first go through the cell status. When we talk about the cell status, here I'd like you to know 2 of our 3 cells, right, erythrocytes, leukocytes, and platelets. 2 of these 3 cells aren't even considered complete or true cells. Now, which 2? Erythrocytes being number 1. And the reason why is erythrocytes have no nuclei nor organelles. What we're going to see inside of erythrocytes is basically they're bags of hemoglobin, you can think of. They are bags of hemoglobin, and they're packed with hemoglobin. Second are platelets. Platelets, when we talk platelets are cell fragments. They are cell fragments. Fragments meaning pieces of another cell. You'll see, we'll talk in a few minutes. Once upon a time, there was a cell. This cell's name was megakaryocyte. The megakaryocyte then eventually basically breaks apart. And when it breaks apart, it gives rise to a whole bunch of, you can see, cell fragments. And there then we have now platelets formed. Next then we have, we'll see basically the actual true cells are going to be our leukocytes. Our leukocytes are complete cells. Only leukocytes are complete cells. Next, in longevity, we'll see most formed elements, they survive a few days. Others are going to be. So when we talk longevity, we'll see some will have a survival of a few days, while others will last much longer. Now, the third characteristic I'd like you to know about in relation to these formed elements is that they're amitotic. Most of them don't divide. They're going to come from stem cells. We'll see the stem cells, the cells basically that'll give rise to basically these immature cells that'll eventually give rise to these mature cells. Now, first here, let's go through and let's talk about our erythrocytes. Let's check out our erythrocytes. So here we can appreciate a couple of images of some nice erythrocytes. Now, when we talk erythrocytes, here we can see, first let's talk their structure. Their size and shape. They're shaped like miniature donuts. They're shaped like biconcave discs. Biconcave because you can see here on both sides. Biconcave discs. Flattened discs, you can think of. Flattened discs with depressed centers. These cells are small cells. They have about diameter of about 7.5 micrometers, as we can see. They have a diameter of about 7.5 micrometers. And when we talk about the lifespan then of erythrocytes, their lifespan is anywhere from about 100 to 120 days in adults. And when we talk infants, it's anywhere from 80 to 90 days. Now when we talk about organelles, we said there's none. You have this plasma membrane. These cells are anucleate. And they're basically just bags of hemoglobin, as we described. Bags of hemoglobin. So let's look at the proteins that we're going to find inside of here. And the first protein is hemoglobin. When we talk hemoglobin, hemoglobin is a red blood cell protein. It's an RBC protein that functions in gas transport. It functions in gas transport. And here we're going to go through and we'll talk about hemoglobin in a few minutes. The next protein I'd like you to know about here is spectrin. Spectrin is basically a network of proteins that maintain the erythrocyte's shape. That maintain the erythrocyte's shape. So if we don't have these proteins there, they cause the cell to be misshaped, not the same shape as we saw here. And now when it doesn't have that shape to it, then the body basically is, the cell's in danger. The body's going to want to attack that cell and remove it from the body then as well, right? So lots of things can happen then with that as well. So spectrin, very important protein. Next, then let's go through and let's talk about another component you're going to find inside these cells are going to be antioxidant enzymes. And they're going to help basically rid the body of harmful oxygen radicals. It'll rid the body of harmful oxygen radicals. And they mostly function to maintain the plasma membrane. Next then is the function. The function of our red blood cell or erythrocyte is that it's involved in transporting respiratory gases, and we're going to talk about oxygen and CO2, right? In human physiology, human anatomy, that's really what we're concerned with. And then when you get to other programs, you may move to RT or you may move to nursing. They'll teach you then about other gases that we want to that might be of interest. Nitrogen is going to be that one. Next, then we have hemoglobin. So let's talk hemoglobin. Hemoglobin is a protein. It's a protein that makes red blood cells red, and it binds easily and reversibly with oxygen. Hemoglobin is made up of heme and globin. So when we talk globin, globin is a protein. Globin is going to consist of 4 polypeptide chains, as we can see here. 4 polypeptide chains. Right? These are all biology terms, polypeptide chains. Okay? Each polypeptide chain we can see here is responsible for binding to a ring-like heme group. Now we talk globin chains. Here in adult hemoglobin, we can see we have 2 alpha globin chains and 2 beta globin chains. And you'll learn about hemoglobin F, fetal hemoglobin, and it's different. And why then we use it, for our patient's advantage, even when we need. So you got to learn the basics. You got to know this stuff to be able to learn that more complicated stuff. If you don't know this stuff, there's no way you'll get a comprehension of that other stuff then. So you want to get this well. So next thing is heme. When we talk heme is a red pigment, and it bears an atom of iron. It bears an atom of iron to which oxygen can bind reversibly. I want to also tell you if carbon monoxide, another gas you're going to be learning about, is in the picture. If they're both in the picture, oxygen doesn't get to take the seat. Carbon monoxide takes the seat. So carbon monoxide is a very poisonous gas. So it says basically, hey, I get shotgun, I'm riding shotgun. You get out of that seat. So hemoglobin, we can see here, and oxygen again is going to bind to here, and you see why it's important. And we even want to replenish this iron in patients as well. But replenishing that iron is not anything easy to do. A lot of other things come into play as well. Constipation is one problem that can come into play. So then we got to switch different types of iron and help with that. But it's really important. You can see there. Next, let's talk about leukocytes. Let's talk about our leukocytes then. So we're good with our red blood cells. You got the basic idea there. Now, when we talk leukocytes, we'll talk in a few seconds. But here you could see the whole pathway of basically production of erythrocytes. So here you can see these erythrocytes, again, they themselves are amitotic because look at here, they came from this hemocytoblast, which was a stem cell. That stem cell became a proerythroblast, a committed cell, which then undergoes this developmental pathway. Here you can see what happens. It becomes an early erythroblast, a late erythroblast, a normal blast, and then it loses its nucleus, and then it becomes a reticulocyte, and then it becomes an erythrocyte. So we'll look at the development of basically the white blood cells as well. So we talk leukocytes. Leukocytes are white blood cells. White blood cells. They're the only formed element that is a complete cell, as we've described, because they have organelles and nuclei. And their lifespan can range anywhere from 13 to 20 days. 13 to 20 days. Now, some characteristics here in relation to our white blood cells, the leukocytes, is first that they exhibit what we call diapedesis. They exhibit diapedesis. They have the ability to slip out of the capillaries, our blood vessels. They have the ability to slip out of our blood vessels. Also here, when we go through and we talk about our characteristics in relation to these white blood cells, they exhibit positive chemotaxis. So here, what we're basically saying here is when a chemical trail is left by molecules that are released by damaged cells, well, that trail gets followed by these cells. So they can pinpoint areas of tissue damage and infection. And then that allows them to gather there in large numbers to help destroy the foreign substances or even remove the dead cells. And when they basically are moving, they exhibit amoeboid motion basically there as well. Now, when we go through and we talk about our leukocytes are going to get divided up into two major categories. They'll get divided up into granulocytes and agranulocytes. We saw that here. And here they actually mentioned each type, and we'll look at that as well. So you can see the same thing here. It tells you the population of each type. We're going to go through those, so you're going to have to know those as well. Here you can see the different types. Here you can actually see the two different categories in two pictures. So granulocytes, agranulocytes. Let's talk granulocytes. Granulocytes, they contain these obvious membrane-bound cytoplasmic granules. So hence the name granulocytes. First we have neutrophils. Neutrophils are the most numerous white blood cell. They make up about 50 to 70%. When you talk about their structure, they stain a pale lilac. Their nuclei are going to consist of 3 to 6 lobes. Those nuclei are multi-lobed nuclei. Anywhere from 3 to 6 lobes to their nuclei. Also known as polymorphonuclear cells because of the nuclear variability. Now their function is to phagocytize bacteria. Their function is to phagocytize bacteria. So they're going to be involved in bacterial infections. So you have to be able to understand and diagnose that from the questions that could come according to that for your exam. Eosinophils. Eosinophils, they're going to account for about 2 to 4% of all leukocytes, of all white blood cells. Eosinophils, their structure, they have a bilobed nucleus. They have bilobed nuclei. You can see that here very nicely. One lobe, and here we have the other lobe, bilobed nuclei. And they have red cytoplasmic granules, you can see. They have red cytoplasmic granules. When you talk about their function is to kill parasitic worms. It is to kill parasitic worms. And they also play a role in allergy and asthma. They also play a role, they're involved in allergies and asthma. So if you have a patient depicting basically these symptoms of allergies and or asthma, you can confirm that with an increased quantity in a blood check of eosinophils. Next are basophils. Basophils are the least numerous, anywhere from 0.5 to about 1% of all leukocytes will be basophils. Now, when you talk about their structure, they have bilobe nuclei, large purplish black cytoplasmic granules, which contain histamine. So their functions are going to include that, number one, they release histamine. Histamine is a vasodilator. Right, so you got to make sure you read about histamine and understand histamine. And also other inflammation mediators as well. Also, we're going to find here heparin. So make sure you read about heparin, understand heparin, and how it functions as an anticoagulant. So from our granulocytes now we can move over to our agranulocytes. Agranulocytes now, white blood cells that lack visible cytoplasmic granules. The nuclei here, they're typically spherical or kidney-shaped. Now, when we talk about the structure of our lymphocytes, so first type of agranulocyte we'll talk about are going to be our lymphocytes. When we talk about lymphocytes, I want you to know first that the lymphocytes, they're going to account for about 25 to 33% of leukocytes. Lymphocytes, we have two types. Now, we talk about two types. Here we're going to see we have B lymphocytes and we have T lymphocytes. Our B lymphocytes, they go on to become plasma cells. They will go on to become plasma cells. Plasma cells then are going to be responsible for producing antibodies. They produce antibodies. You'll learn about antibodies in microbiology. Antibodies protect us against antigens, against foreign antigens. Next in here, we have T lymphocytes. T lymphocytes, they're going to function in immune response. They function in immune response by acting against virus-infected cells and tumor cells. So when we talk lymphocytes, they mount immune responses by direct cell attack or via antibodies. And they have, you can see, spherical nuclei, kind of indented on one side sometimes. Next in our monocytes. When we talk monocytes, monocytes are going to account for 3 to 8% of our white blood cells. They are the largest leukocyte that we'll have and we'll see. And when we talk about their structure, they have U or kidney-shaped nuclei. And they have a gray-blue cytoplasm. And their function here, when we talk function, so they're involved with phagocytosis. They develop into macrophages in tissues. And these monocytes are important in chronic infections. I want you to go through and read about each of these cells and understand each of these cells in greater detail. Okay? So here when we talk monocytes, they're going to be involved in infections like TB, with mounting attacks against viruses and intracellular parasites. And we talked eosinophils. Eosinophils, we talked about also killing parasitic worms. They kill parasitic worms as well, and they play a role, I told you, in allergy and asthma. So I want you to go through and I want you to really understand each of these cells in greater detail. Read through each of them and get the full-on idea understood with each of these cells. Next, then we have our platelets. When we talk platelets, their structure, they're discoid cytoplasmic fragments that contain granules. They stain a deep purple, and their lifespan is anywhere from 8 to 9 days. When we talk about their function is that they seal small tears in blood vessels, and they're involved in blood clotting. And we saw how they came from basically, I told you how they came from these megakaryocytes. So here we can appreciate those fragments. So here we can see now the development of basically these leukocytes. Here when we go through and we talk about the development of formed elements, we saw with hemocytoblasts and erythrocytes basically, right? And then the hormone involved there, you've got to know that. And then the process of basically production of red blood cells and the maturation of red blood cells is erythropoiesis. Erythropoiesis. And I mentioned the hormone EPO erythropoietin, right? You had to know that from endocrine chapter. Next, then here we could see leukocytes. Leukocytes then, you can see are going to come from hemocytoblasts. These hemocytoblasts are going to give rise to myeloid stem cells and lymphoid stem cells. Then these lymphoid and myeloid stem cells will give rise to their respective cells. The myeloid stem cells, you can see, give rise to these committed cells, the myeloblasts and monoblasts. Now, each of these myeloblasts and monoblasts are going to give rise to promyelocytes and promonocytes. Then each of them will give rise to their respective cells. You can see promyelocytes will go on to become eosinophilic myelocytes, basophilic myelocytes, neutrophilic myelocytes, which then continue to become band cells, and then they become their mature granular leukocytes. And then when we talk promonocytes become monocytes, and then here you could see some will then go on to become, as we said, giving rise to macrophages and whatnot. And here's same thing with lymphoid stem cells, lymphoblasts, prolymphocytes, and then lymphocytes. And then some will go on and basically go through anticlonal, or basically give rise to antibodies and whatnot, become plasma cells, and then give rise to antibodies and whatnot. So you could see that there very nicely. Next, then platelets, we said cytoplasmic fragments of megakaryocytes. Okay, so you could see the same thing here now. They exhibit a blue staining kind of outer region and an inner region that contains these granules that stain purple. And these cells are essential for clotting. And here you can see hemocytoblast becomes a megakaryoblast, which becomes then a promegakaryocyte, which becomes a megakaryocyte, which then gives rise to, we can see platelets.